Gregor Johann Mendel was an Austrian monk and he studied the inheritance of traits in pea plants. 1822 to 1884, Mendel developed two laws of inheritance using the statistical data he collected. Piscium sativum, the garden pea plant, was used by Mendel for his experiments. Mendel's procedure: he removed the anthers of the flower he termed as male flower in the first instance and named it as female flower. Then he took pollen from the new male flower and applied on the stigma of the new female flower. After fertilization he collected the seeds and planted them again. Then he observed the relevant feature of the progeny. Mendel carried out the reciprocal procedure too. He got same results for the reciprocal test too. Mendel carried out crosses on 7 traits of pea plants. Height of the plant: tall plants, dwarf plants. Color of the flower: white flowers, purple flowers. Position of the flower: axial flowers, terminal flowers. Color of the seeds: yellow seeds, green seeds. Shape of the seeds: round seeds, wrinkled seeds. Color of the pods: yellow pods, green pods. Shape of the pod: inflated pods, constricted pods. How Mendel identified pea plants: he observed the transmission of a trait along several consequent generations by self-crossing the plants to identify the pea plants this way. Tall plants. Tall plants. All the daughter plants were tall. Then he self-pollinated daughter plants. All the daughter plants obtained by self-pollination were also tall. He carried on this process along several generations. If he got only tall plants along several consequent generations, he termed the mother plant as a pure tall plant. Pea tall plant. Then he crossed dwarf plants using the same procedure to obtain pea dwarf plants. Dwarf plants. Dwarf plants. All the daughter plants were dwarf. Then he self-pollinated daughter plants. All the daughter plants obtained by self-pollination were also dwarf. If he got only dwarf plants along several consequent generations of self-crossing, he termed the mother plant as a pure dwarf plant. Pure dwarf plant. Likewise, he carried on the same procedure for all the 7 characteristics he used in his breeding experiments to get pure plants. Mendel then crossed a pea tall plant and a pea dwarf plant. Pea tall plant. Pure dwarf plant. All F1 plants of this cross were tall. He self-pollinated F1 members and he got 2 phenotypes for F2 generation with 3:1 ratio. Mendel's results can be clarified using simple genetic diagrams. Relevant letters are used in these genetic diagrams to indicate characteristics. Pea tall plant, pure dwarf plant, according to Mendel. Each heritable character is determined by 2 heritable factors. During the formation of gametes, these 2 factors for a heritable character are separated and get into each of the gamete formed. Gametes. Random fertilization of gametes. Tall plants, self-pollination of F1 plants. 3 tall plants. 1 dwarf plant. Reginald Punnett 1875–1967. Punnett's squares introduced by Reginald Punnett at a later period, can also be used to explain the behavior of alleles in genetic crosses better. Let's clarify the Mendel's results further. By a Punnett square, Mendel stated that physical traits are inherited by factors. Entering of factors into the gametes, and their random fertilization to produce an offspring. F1 generation produced by fertilization of gametes. Mendel got 2 phenotypes as tall plants and dwarf plants with 3:1 ratio for the F2 generation. Mendel's first law is known as law of independent segregation. The factors he introduced were identified later as alleles located in chromosomal DNA. Chromosomes. Homozygous alleles. Heterozygous alleles. Let's clarify the Mendel's results further, with the knowledge of chromosomes and alleles discovered by other scientists later. PO plants bear homozygous alleles. Homozygous dominant alleles. Homozygous recessive alleles. When 2 PO plants are crossed, all the members of progeny consists of heterozygotes. PO tall plant. Pure dwarf plant. Chromosomes. Alleles getting into gametes independently, and random fertilization of gametes. F1 generation. All plants of F1 generation were tall plants, and all the plants were heterozygotes. Self-pollination of F1 plants, heterozygous tall plants. Alleles getting into gametes independently, and random fertilization of gametes. F2 generation. 3 tall plants. 1 dwarf plant. Mendel got 2 phenotypes as tall plants and dwarf plants with 3:1 ratio for the F2 generation. Mendel stated that he got this type of results for both F1 and F2 generations, due to this independent segregation of alleles which occur in gametogenesis, and he produced his first law on the basis of this phenomenon. Mendel got 2 phenotypes with 3:1 ratio approximately, for F2 generations of all the monohybrid crosses he did. Transmission of only one characteristic is observed in monohybrid crosses. Mendel used his results of monohybrid crosses to introduce his first law. Summary of Mendel's results of monohybrid crosses. Trait. Dominant phenotype. Recessive phenotype. Number of plants with dominant character. Number of plants with recessive character. Height of the plant. Tall plants. Dwarf plants, 787 tall plants. 227 dwarf plants. Purple flowers. White flowers. 705 purple flowered plants. 224 white flowered plants. Color of the seeds. Yellow seeds. Green seeds. 6,022 yellow seeded plants. 2,001 green seeded plants. Shape of the seeds, round yellow seeds. Wrinkled yellow seeds. 5,474 yellow round seeded plants. 1,850 yellow wrinkled seeded plants. Color of the pods, green pods. Yellow pods. 428 plants, with green pods. 152 plants, with yellow pods. Shape of the pod. Inflated pods. Constricted pods. 882 plants, with green inflated pods. 299 plants, with green constricted pods. Position of the flower. Axial flowers. Terminal flowers. 651 plants with axial flowers. 207 plants with terminal flowers. Ratio. .84 to 1. 3.15 to 1. 3.01 to 1. 2.96 to 1. 2.82 to 1. 2.95 to 1. 3.14 to 1. Mendel got two phenotypes with 3:1 ratio approximately for F2 generations of all the monahybrid crosses he carried out. Mendel used these results to produce his first law. Mendel's first law. Each heritable character is determined by two heritable factors which are known as alleles. During the formation of gametes, these two factors for the heritable character are separated and get into each of the gamete formed. Hope to discuss Mendel's second law by our next video. Thank you for watching. Subscribe for more such videos. Subscribe for more such videos.